In this video, I'm going to show you how to complete the qualitative analysis part of SOP2. In the first year labs, we focused entirely on quantitative analysis. So that's analysing a sample in order to determine exactly how much of a component of that sample is present. But there is another kind of chemical analysis called qualitative analysis. And when you analyse a material qualitatively, what you're asking is, what is it actually made of? What can we detect in this sample? And in this part of SOP2, your job will be to qualitatively analyse a GCMS dataset. Now this dataset is actually turpentine dissolved in hexane, but that hexane contained a lot of alkane contaminants. And in this video, I'm going to show you how to find these alkane components in a complex GCMS sample. And then your task will be to use the same approach in order to identify the monoterpenes in this dataset, a different class of chemicals. To complete this task, you'll be using a piece of software called AMDIS. AMDIS stands for Automated Mass Spectral Deconvolution and Identification System, and it was developed by part of the US government called NIST. It's very commonly used for GCMS data processing around the world, and the great benefit to you guys is it's free. There are two ways you can get hold of AMDIS. If you're working on a Windows computer, you can download it by following the link here. At the bottom of that chemdata.nist.gov page, you'll find the download link. Alternatively, if you're working on a machine that you can't install Windows software on, like a Mac or a Linux or a machine which is tied down, you can access it via the NTU Virtual PC. So go to my.ntu.ac.uk and then find the Virtual PC link. I believe the tabs move around. But then once you've opened up the Virtual PC, you'll find AMDIS is in the software hub and you can start it from there. Before we go on to look at how to use AMDIS itself, it's important to remind ourselves about the 2D nature of GCMS data and also some important nomenclature, like the total ion chromatogram and the extracted ion chromatogram. Normally, when you view GC or LC-MS data, the screen that you're looking at will have a display showing a chromatogram and a display showing a mass spectrum. But that kind of view hides the essential two-dimensional nature of this kind of data. So let's remind ourselves what's inside a GC or LC-MS dataset, in this case by using a GC file. During an analytical run, the instrument is collecting one mass spectrum after another, and this display shows the data as it's collected. And once it's collected, each mass spectrum is added to a table. In this particular view, the most intense peaks are shown as red or white dots. So in this dataset, each row corresponds to the mass spectrum that was recorded at a particular time in the chromatogram. So now we've completed collecting our big dataset, what sort of different views of the data can we get from within it? So one of the most common views is called the total ion chromatogram. And we get that by collapsing this two-dimensional data in this direction, basically summing up each row and plotting it as a chromatogram. So let's consider this really intense spectrum that was recorded just before 10 minutes. If we sum up all the intensities of all the masses in that mass spectrum, that's what gives us the high value that corresponds to this peak in the total ion chromatogram. Another less common view is called the total mass spectrum, and that's what happens if we collapse the 2D dataset in the other direction, basically summing up all the intensities in all mass channels irrespective of when they were recorded in the chromatogram. Now, having collected our large 2D GCMS dataset, there are two common ways we can extract information out of that. One of them is known as the extracted ion chromatogram, or XIC, and another one is the extracted mass spectrum. So let's look at how we derive those from a 2D GCMS dataset. I've set this program up so that as I move the cursor, it grabs two slices out of the GCMS data. I've moved the cursor through the two-dimensional dataset to sit at the coordinates of mass to charge 93 and retention time 9.37 minutes. So with the cursor in this position, the chromatogram now shows how the intensity for the ion mass to charge 93 varied throughout the chromatographic run and ignores all the other ions. And as we've extracted just that single ion out of all the data, this is known as an extracted ion chromatogram. But we can also take a slice through the data in the other direction, in this case to recreate the mass spectrum that was recorded at the time of the cursor. And using the same nomenclature, this could be known as an extracted mass spectrum. And as I move the cursor around inside the dataset, you can see I can extract other ion chromatograms with peaks in different positions and other mass spectra. But to recap, a GC or LC-MS dataset is really a two-dimensional table of data. And from that we can extract ion chromatograms that show how the intensities of individual ions varied as a function of time during the chromatography. And we can also extract mass spectra that showed what the complete signal looked like at a given retention time. To load the dataset into AMDIS, go to the File menu. Then choose Open. The file you need for this task is available from now, so download it to a folder on your computer or to the Downloads folder in the virtual PC, and then navigate to it using this dialog box. It's important that this particular kind of GC file is actually a folder, and when you look at it through Windows Explorer, it will appear like a normal folder, just with .d at the end of the name. So when you download the file from now, unzip the contents, but make sure you keep it within a folder that ends .d. AMDIS will then open the file and display the TIC, and in this case an extracted spectrum. The window you see in AMDIS may look slightly different, but don't worry about that just now. Now this is quite a complicated sample, and there's a lot of peaks in there, so let's zoom in to a small part of the chromatogram so we can see what's really going on. In AMDIS, to zoom into an area, just click and drag over the area of the chromatogram you want to zoom into. To view the extracted spectrum from any retention time, simply click in the chromatogram at that retention time, and the corresponding extracted mass spectrum will display in the bottom panel. Currently, the chromatogram panel is only showing the total ion chromatogram. So how can we get extracted ion chromatograms out of datasets in AMDIS? All you need to do is click on the corresponding mass in the mass spectrum panel. And there doesn't even need to be a peak in the spectrum there, you can click it anyway. So I clicked on the peak at mass 57, and now I have the extracted ion chromatogram for mass 57 showing in the chromatogram view. Now you can see there isn't actually a peak at 59 in the mass spectrum, but I'll click there anyway to produce the extracted ion chromatogram for mass 59. So now we can see the extracted ion chromatograms for mass to charge 57 and 59 superimposed on the TIC in the chromatogram panel. And what this shows us is that there are several components in this region that produce a fragment 57 under electron ionization conditions. But there's only two that are producing a significant fragment at mass 59. This region of the chromatogram is quite interesting, so let's have a closer look at it. As compounds elute from a GC column in a GCMS, they're ionized, and any fragments and ions produced by them should be detected at the same time by the mass spectrometer. In this spectral region, there's a peak coming from ion 57, and there's also a peak from ion 59. But if you look at the positions of the tops of those peaks, they're not at the same retention time. So these must be ions coming from different components that are coming out of the GC column almost at the same time. If we extract the mass spectrum in the dataset that corresponds to the retention time of the 57 ion, this is what we get. If you look in the chromatograms, you can see that the component that produces fragment 59 has a long tail, and the component of 57 is coming out in that tail. And sure enough, if we look in the mass spectrum, we can see both the ion 59 and 57 at the same time, even though we know these must come from different components. So looking at all the other peaks that are in the mass spectrum, how can we tell which peaks come from the component that gives fragment 59 and which ones come from the component that gives 57? And that's where AMDIS can come in and help us. What AMDIS does is it goes through the whole 2D GCMS dataset and finds all the ions that have the same retention time and groups them together as a component, and then it removes them from the dataset and looks for the next component. And it keeps on going until it's found all the components it can in the dataset it's been given. So to start this process, we click the Run button. That process was very fast, but then we're only looking at a tiny proportion of the total GCMS dataset. AMDIS only processes the parts that are visible in the chromatogram. You'll also note that AMDIS has added an extra couple of windows to the middle of the display, but ignore them just now and just concentrate on the top chromatogram panel and the bottom mass spectrum panel. Above the chromatogram panel, you can see AMDIS has added a series of little blue triangles. Each one of these triangles corresponds to the retention time of a component it thinks it's detected. If you click on any one of those triangles, it will display the extracted mass spectrum at that time and also what it believes is the mass spectrum of that component in the mass spectrum panel at the bottom. So first of all, let's look at the component that gave us the fragment 59. In the mass spectrum panel, you can now see that there are two mass spectra being shown. The mass spectrum in black is just the extracted mass spectrum at that retention time. So that could include ions from multiple different components. But the mass spectrum in white is the spectrum that AMDIS thinks corresponds to just that component. So basically it's subtracted all the other potentially co-eluting ions away. So you can see that the ions that come from the component that gave us fragment 59 dominate the mass spectrum at this retention time. But it's more interesting if we look at the ion which gave us the fragment 57. So to view that, we'll click on its little component tick. And now, in the mass spectrum, we can quite clearly see that the 59 ion does not come from the same component that gives us the 57 ion. This process of extracting a component mass spectrum out of overlapping signals is called deconvolution and is the D in AMDIS. And the great thing about AMDIS is that having produced this pure mass spectrum of whatever component we were looking for, you can now search that against a library, if you have one installed, in order to try and identify it. But we're going to be using AMDIS for a different task, so let's look at that. In this exercise, we're going to use extracted ion chromatograms generated in AMDIS in order to identify all members of particular chemical families in the data. The kind of ionization used in GCMS is electron ionization, and we have to remember that's a hard ionization source, which means that it commonly produces fragments as a result of the ionization process. Now usefully, these fragments aren't random, but are actually a result of the structure of the molecule being ionized. So if you have a group of compounds that you're interested in that are all structurally related, then you'd expect them to produce similar fragments under electron ionization conditions. What we're going to do in this exercise is use that knowledge to help us find a group of structurally related compounds in our GCMS dataset, in this case alkanes. I found these mass spectra of long-chain alkanes by looking them up on the NIST chemistry webbook. You should have a search for that on Google. Now, although these are all different molecules, you should be able to see that they all produce the same set of common fragments as a result of electron ionization. So these ions at 29, 43, 57, 71, and 85 form a kind of signature that we can use to identify long-chain alkanes under GCMS conditions. And in the case of alkanes, it's easy to see where these fragments come from. They're simply fragments along the long-chain alkane backbone, like this. Using octane as a simple example, if we were to imagine that alkane backbone can be fragmented between any pair of carbons, we can easily see how the sequence of fragment peaks we see in the mass spectra can be derived from this molecule. So now let's look and see how we can use extracted ion chromatograms in AMDIS to search for alkane-like molecules in our GCMS data. Back in AMDIS, the first thing we'll need to do is zoom out to view the whole GC dataset. So right-click in the chromatogram display and choose Unzoom All. We only used AMDIS to find components in a small region of the dataset before, so now we've zoomed out to show the full data, we'll need to hit Run again to deconvolve the complete dataset. So AMDIS thinks there's 248 components in this dataset, and we could go through each one of those manually to check and see if it looks like it contains the signature ions of an alkane. But I'll show you there's a much faster way we can filter down to a smaller subset of ions using extracted ion chromatograms. The first step is to get rid of the current extracted ion chromatograms so we can replace them with the ones needed for alkanes. To delete an extracted ion chromatogram in AMDIS, you just have to click on the label. The mass range in this GC dataset doesn't go far enough down to see the ions at 15 or 29. So we'll be looking for alkane ions at 43, 57, 71 and 85. And to add those XICs, I just click on a peak, or at least that position, in the mass spectral display at the bottom of the screen. Now, as we discussed before, this is quite a complicated dataset, and we can't really see what's going on. So let me just zoom into a smaller region so we can see the detail better. In this small spectral region, there are several components that I'm pretty confident are alkanes, and some other components that I'm pretty confident are not alkanes. So based on the information on this screen, how do I know what components are or are not alkanes? For these 5 components, I'm sure that none of them are alkanes. And I know that because none of these components are associated with peaks in the extracted ion chromatograms for the alkane signature ions. But in this region, all of these 5 components almost certainly are alkanes. And I can predict this with confidence because all of the alkane signature ions go up and down together for that component. But in the same spectral region, these 2 components are not alkanes. And I know that because they're not associated with peaks in the extracted ion chromatograms for the signature ions associated with alkanes. Now, of course, I don't know the identities of the alkanes I've found. I just know that the peaks in this region are alkanes. In order to identify them, I need to match the mass spectrum against a library, or collect a verified standard sample and check that it has the right retention time and produces the right mass spectrum. But when you're screening samples, it can be very useful to be able to quickly find members of particular chemical classes. In this example, I've shown you how to find alkanes, and in the SOP you use the same approach to try to identify the monoterpenes in this sample. So the same as I demonstrated here for alkanes, you should find a list of monoterpenes, then look up their spectra on the NIST chemistry webbook, and by looking at those mass spectra try to identify fragmentines that are common across many monoterpenes. Then you make their extracted ion chromatograms visible in AMDIS, and look to try to find peaks where many of those fragmentines go up and down at the same time. In the next slide I'll show you how to get the retention time and the peak area of the compounds you then identify as monoterpenes. So as I said, how do you get the peak areas and retention times of components from AMDIS? Actually, AMDIS makes this quite easy. All you have to do is click on the relevant component triangle, and then the peak area and retention time are shown over here. And then to view the data for other components, you simply click on their tabs. The retention time for a selected component is always the highlighted one in the list. You could actually use these peak areas to generate the calibration curves from standards, and then to use that calibration curve to quantify the components in unknown samples.